The first step when you build a house is having very solid foundation. And to me, the foundation of the house is the supply of your clinical phase 1. You have to see that as the foundation of the house. Here, you cannot make mistakes. And there are mistakes I have seen during my career. When they are done at that stage, they cannot be repaired. A typical example of this type of mistake could be the clonality of your cell banks. Developing cell bank has been a topic for years, but also the technology and the expectations from regulators have also evolved. So you cannot develop a cell bank today as you were doing it 10 years ago.
Many biologics that fail in late-stage development share one fatal flaw: a weak CMC foundation laid years earlier. Henri Kornmann has spent 2 decades developing biologics the right way, from Merck to Biogen to leading Ferring's first gene therapy approval for bladder cancer. Today, he reveals why your clonal cell line selection, analytical methods, and early characterization decisions determine whether your therapy reaches patients or collapses under regulatory scrutiny. If you're developing biotherapeutics, the choices you make today will haunt or save you at commercial scale. Let's dive in. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brohlman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Henri. It's good to have you on today.
Hello, David. It's good to be with you and with the listener to your podcast today.
Henri, share something that you believe about bioprocess development that most people disagree with.
That's a difficult question. I think there is something very important for people to understand in drug development in general, but this is even more valid for all the CMC aspect of the drug, is that a CMC program is like building a house. You need to start at the beginning of your program at preclinical stage and at, let's say, clinical phase 1. You need to start with very, very solid foundation. And from those foundation, as you progress with clinical phase 2 and 3 and then request for approval, this is like adding floors and rooms in the house. And I have been in this industry for 20 years, mainly in big and mid pharma, never working for startup, but I have perform a lot of due diligence. And when I have to due diligence a program, I often realize that colleagues and in startups have difficult to understand that. And of course, I mean, they have limited financing and they need to go fast. But in that environment, they often forget about some of the fundamental of CMC that are critical.
Yeah, I share your observation, Henri. As a consultant, I speak to a lot of startup founders, and that's exactly what I noticed, that a lot of startup founders have next to nothing, or in some cases, little knowledge about what CMC entails. And also, I like your way of looking at it, building a house, and you need to lay a solid foundation. And that's also the reason I'm very excited about today's podcast, because we're going to look at how are we going to build build that house that is gonna endure and not gonna fall eventually. But before we do that, Henri, take us back to the beginning and tell us what sparked your passion for biotech and what were some interesting pit stops along the way.
My career is rather simple. If you draw a line between CMC development and GMP manufacturing, during my career, I constantly jump from one side to the other. For instance, I started my career at Merck as junior CMC development scientist, being specialized in downstream processing. But then I jumped and I became Lean Six Sigma black belt, improving GMP manufacturing process. And then ultimately progressing into being responsible for the GMP manufacturing in a multi-product facility based in Switzerland. And then I moved back to CMC development, developing biosimilar. Then I made a 3 years excursion out of biotech working for medical device, working for an American company called Medtronic. But of course, I miss biotech. So after 3 years, I jumped back into tech ops, working for Biogen, being part of this fantastic facility that they have in the middle of Switzerland, close to Solothurn. And then ultimately, and this is where I am today, I was called to join Ferring. Ferring is mid-pharma company, and I joined Ferring to develop the CMC development side for Biologics.
Tell us about this last part, because you were involved in the approval of Ferring's first gene therapy. Tell us more about that, and then also tell us how that experience reinforced the importance of getting your CMC foundation right.
These programs, this is a gene therapy helping patients fighting with bladder cancer. This therapy has been now approved by FDA. Ferring gets involved in that product at the very end of clinical phase 3. And then the mandate was to take it from there and to bring it to BLA. And it's a very good program. It has been developed by one of our sister company based in Finland called Vector. This gene therapy program is called Adstiladrin. It's gene therapy helping patients fighting for bladder cancer. And of course, one of the first things you do is review the program and you assess the various CMC work package to see if they are complete or if there are some gaps. Because of course, when you go into registration, you submit your dossier and you get questions. I think every development program has CMC challenge, and that one was not different than others. So we started to close some of those gaps. But it was interesting because there were some gaps that were difficult to close because we were late in the program. And actually, I was saying before that all programs have challenges. There has been this review from FDA mid of last year, I think in July, where they published the list of the complete response letter starting from 2020. So all the complete response letters starting, most of the complete CRL, the complete response letter starting from 2020. And people started to analyze the cause and why the program were not approved the first time. And actually 70% of the CRL were having a CMC challenge. So I think this indicates to colleagues working in CMC that this is extremely important, because you don't want to arrive there and having your program reject out of CMC topic. There has been another FDA analysis. I think it's an older one from 2020 that was not on the CRL, but more on the IND. And that study showed also that a significant amount of those IND, around 10%, they were not allowed to proceed to clinical phase 1, also because of CMC issues. So at the end of the development and at the beginning of the development, CMC can cause delay, increase cost and delay in program if it's not properly done.
You're making an excellent point, and I just want to reiterate that because this is such an important statement. And also, I want to raise awareness because as I'm having conversations with people who want to go into an IND filing, it's so key to understand that you need to get these things right early on. And as you said, Henri, if you make wrong choices or you have some gaps, these can become very costly, especially at the later stage of your program as you're getting closer to your BLA. And that's the very topic of today, to help biotech scientists understand, okay, this is important, but also have a strategy and have tactics to make better choices. So let's go into your analogy of building this house. Tell us, where do you start? What is the foundation? What do we have to look at? And then we'll go step by step.
I like this analogy very much. I'm using all the time when I mentor the next generation of CMC scientists and the next generation of CMC leader, because I think it's quite self-explaining. So maybe the first step when you build a house is having very solid foundation. And to me, the foundation of the house is the supply of your clinical phase 1. Okay, you have to see that as the foundation of the house. Here, you cannot make mistakes. And there are mistakes I have seen during my career. When they are done at that stage, they cannot be repaired. A typical example of this type of mistake could be the clonality of your cell banks. Developing cell bank has been a topic for years, but also the technology and the expectations from regulators have also evolved. So you cannot develop a cell bank today as you were doing it 10 years ago. I have faced, for instance, during due diligence programs where you could not demonstrate the clonality of the cell bank. So what do you do? What do you recommend to your business development department when you realize that it has not been properly done? And that's the analogy with the foundation. Another example of one of those basic things linked to the cell bank, but also linked to the material you will produce for phase 1 is how do you store it? I mean, it has to be stored according to the good distribution practice. And I have seen example where people have developed properly a cell bank and then they place it in a cryoconservator that is not according to the GDP rules. And so what do you do? Again, when you observe that and you have to make a recommendation, do we buy or not this program when you're part of a due diligence? What do you do? It's a challenge, right?
What about raw materials when you're creating your cell bank? I think that's another critical aspect, right?
You are totally right. This is a key aspect where you need to make sure you have collected all the proper documents about your raw material. I mean, something as basic as a TSC, BSC statement in one of the material you have used to create your cell bank. 10 years down the road, when we are at filing, you can have questions and it can be a challenge. A major question you don't know, but it can be questioned. So people have to remember that. So I think the message is there are few things. In a CMC program, there are a lot of things that can be fixed, but there are few things, and that's the foundation that you cannot mess up. I gave some examples. The idea is not to do an exhausting list, but foundations are important. And you were mentioning that you talk with CMC colleagues in startups. They have to be aware of that, because if you miss some of those critical elements that cannot be fixed on the road, it's destroying part of the value of their program.
Besides the cell bank, is there any other aspect that is fundamental in this foundational part?
I'm sure that there are more. There is one I think about is how deep you characterize your substance. My approach or my recommendation is, even if you are short in money and you have to make some decision, do not save money on the characterization at the beginning as part of the foundation, because you don't want to discover something funky in your molecule at later stage. So full state-of-the-art characterization of your drug substance prior to phase 1 will be a recommendation. there has been programs where you realize that you have very high level of misincorporation in your molecule. I mean, this you want to discover rapidly.
Absolutely. So we have now laid a solid foundation. What is the next step, Henri?
The next step is, so you have your foundation, you have supply for your clinical phase 1. You move into supplying clinical phase 2. My approach and my recommendation is not to change things between supply of phase 1 and phase 2. I mean, some people might want to scale up because they believe it will require less batches and maybe saving money on the supply of clinical phase 2. I mean, my approach is usually simpler. You stay with your phase 1 process. Maybe you produce a little bit more batches to supply your phase 2. This is actually not stupid move because with more batch, you start to build your knowledge about your CQA and you have clinical exposure of your potential CQA.
Can I ask you something about this? Because you mentioned CQAs. So just explain what that is. And it leads me to a further question. When do you start thinking about the quality by design approach? Just explain also what are these different things we should think about.
So the quality by design approach has to be embedded in your program starting clinical phase 1. I mean, you need to think about what would be your potential CQA. So CQA, your critical quality attributes, which means the quality attributes of your molecule that may have an impact on the efficiency and the safety of your product. And you need to think about your potential critical process parameter, which are the process parameter that are impacting the potential CQA. So this has to start phase 1 and it has to be part of your IND. Of course, at phase 1, you know very little about your process and you know very little about your product, but there is a lot of things that are published. If you work with, let's say, platform molecules such as antibody, there is a lot of things that are produced, so you can also use that, but it has to be embedded in your IND for phase 1.
What is a typical mistake or pitfall you see with respect to CQAs or CPPs?
I think one of the challenge QPD is identifying CQA. There are ones that are very obvious, but there are ones you don't know. And of course, in the clinical trial you have are not designed to test your CQA. They are designed to test the safety and the efficacy and the dose of your product. So this is a challenge, and here you need history and literature. And the other challenge is the link between process parameter and CQA, which is usually covered much later in the program during your process validation stage 1. But even there, it's also difficult to cover everything. And this is the reason actually why you have a process validation stage 3, what's the regulator called the continued verification. It's because we know this understanding of the process during clinical development is limited. And we will continue to learn down the road when the product is on the market.
We have explored what makes solid ground for biologics development: a robust clonal cell line, comprehensive analytical panels, justified specifications, and why it's essential to define your critical quality attributes early. In part 2, Henri walks us through scale-up challenges, controlling raw materials and impurities, life cycle management, and building post-approval control strategies that actually work. If these CMC fundamentals resonated, leave a review on Apple Podcasts or your favorite platform to help other scientists like you find this conversation. Thank you so much for tuning in today, and I'll see you next time. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.